Thanks, Anna, for the introduction. As you say, I'm Teresa. I'm a bioinformatician at the Bioimage Archive. And in this first talk, I'm going to talk a little bit about what AI4Life has been doing regarding AI-ready data. So usually for AI applications, bioimage datasets tend to be composed of images, but also annotations, which are the answer or the result for a given task. And these images and annotations are used together to train AI models. And once the models have been trained, they can be deployed to analyze new images, and we can get new scientific insights. But despite how important these data sets are for not only training the models, but also for benchmarking them, to validate them, and also to be able to reproduce published results, AI scientists come across lots of different problems when it comes to data. The first problem is that there are not that many data sets in our field compared to other fields. And that is because for bioimages, these annotations tend to be done manually by experts, so that is very time consuming. Despite this, there are many good data sets in the field, but the next problem is that sometimes they are hard to find. And that is because they get published in scattered repositories. So AI scientists need to kind of like keep track of where each one of the data sets is. Another common problem is that there are many different formats, both for bioimages, but also for the annotations. And that is problematic because sometimes it's hard to even read an image in your own pipeline. And another problem is the metadata. So the metadata is all the information that we need to actually make sense of the data. And in many cases, the metadata for these data sets is incomplete. So sometimes it's hard for a scientist to even know what the license the data are under and if they can reuse them. So in many cases, the data cannot be reused. So to try to address some of these issues, AI4Life is a project that doesn't only that what wants to do is to make AI more accessible for life scientists, and for that we are standardizing the way we share AI methods, but also the data sets that are used to train and to validate AI methods. And this is how the Bioimage Archive got involved in AI4Life. And the Bioimage Archive is EMBOL EBI data resource for biological images. We store and serve images from lots of different imaging modalities, as I hope you can see in this image here. So here you can see some of the images that we have in the repository. These numbers here are the accession numbers that we assign to each one of the studies that get submitted in the archive. So if you are interested in any of the images that I saw during my presentation, you could just take this accession number, go to our website and learn more about them. So what is obvious from this slide is that we have different imaging methods here. We have transmitted light microscopy. This is fluorescent microscopy. This is a micro-CT scan, histology. This is an ultrasound image, and this is a photograph of a whole organism, which also shows that the scale of the images that we have in the archive is very large. We have from molecules all the way to organisms. And finally, something else that is different between these images is the purpose that they serve. So here we have some images that belong to developmental biology studies. This one belongs to a biodiversity study. These are from spatial transcriptomics. And this one here is a data set that actually was developed to train AI methods. And this is a type of data set that we are seeing growing more and more in the archive. I think around 15% of our data sets are related to AI. And this dataset was submitted together with annotations. So to the Bioimage Archive, you can submit not only images, but also annotations, such as segmentation masks, as it was in this case, but also bounding boxes, class labels, point annotations, anything really. So you can submit just images. You could submit images under corresponding annotations, like if it was in this case. And you could also submit just annotations for a pre-existing data set. So what we aim to achieve with the Bioimage Archive is to enable the reproducibility of public scientific results. And most of our submissions are related with scientific publications. but we also serve reference data sets that are useful for different communities. So we have, for example, cell atlases or benchmarking data sets. And finally, we really want to promote the reuse of biological images. They can be reanalyzed and we can gain new scientific insights, but they can also be used, for example, for training. And we know of different bioimages that actually, analysts that use our images for their training. But also they can be used to develop new image analysis methods, including, of course, AI methods. But why would anyone actually submit to the bioimage archive? First of all, you will be contributing to make science more open, and I hope that's something that most people here care about. The same way that we make our code open, we should be making the data that we use open as well. We also be delegating long-term data management, and this is a little bit more of a selfish point, but it's actually a very important point because images are getting larger and larger, so it's hard for labs and for facilities to actually store these images in the long term. So if you submit to the bioimage archive, we will be hosting your images for free, and we do it for life. It has been also published in many different scientific publications that if you make your data publicly available, your papers will get more citations. And finally, you will be enhancing potential for collaboration. So if somebody submits a data set to the archive, then somebody else could use that data set to develop a new image analysis method. And then in turn, the original submitter will benefit from that method that they can use now in their research. And at the core of everything we do is trying to make these images and these annotations fair, which stands for findable, accessible, interoperable, and reusable. In order to be making these images and these annotations fair, which is something that can help to solve a lot of the problems that I initially mentioned, we need to have standards for both the data and the metadata. And the reason why we need the standards is because without them, the bioimage archive will look like this. We will have lots of different files, but we wouldn't know what each one of these files contains or how to find them. By standardizing metadata, we can actually enable search and we can know where each one, what each one of these files contains. But by standardizing the data itself, we can achieve something that is very important when we are talking about images, which is to allow data visualization. And that's something that is not trivial when we are talking about bioimages. And I'm going to discuss both of these points today, starting from the metadata perspective. So when we started AI4Life, there was already a metadata standard for biological images, which is quite well known. It's called REMBI, or Recommended Metadata for Biological Images, and it stemmed from a workshop organized by the community in 2021. So this group of scientists came up together with a model that has several modules. And for REMBI, you will have to input information, for example, about the organism that you are imaging or the cell line that you are imaging, what organ, what tissue, etc. You will also have to explain, for example, how you prepare your sample for imaging, and importantly, how you acquire your images. But when we started AI4Life, something that we didn't have was a standard for annotations. And this is very important because, as I mentioned before, for many different AI methods, annotations are very important. And if we don't have a metadata standard, we don't have a proper way to share them and to make them reusable. So we also organized a community workshop I think it was at the beginning of 2023. And we had data producers, we had AI scientists, we had data management experts, and bioimage analysts. And all together, we came up with the following standard, which is called MIFA. And as you can see here in these different circles, it also has different modules of information that we should provide. For example, an important module is to the annotators module. We want to really give credit to the people that do the hard work of annotating, because as we mentioned before, in many cases that's done manually by experts. So we really need to be able to recognize the people that do this work. Other information that we will have to give for the annotations will be, for example, the annotation type that we are creating. So are we creating tracks or counts or bounding boxes, segmentation masks, etc. And an important module here is the method. The method is about how you created the annotation. So for example, were the annotations created manually or were they created using specific software, and what software you use? Were the annotations created by an expert or were they crowdsourced? What quality control was applied, etc. Then we have other modules, for example, the confidence that we can have on our annotations, the criteria, so the rules that we follow when we were annotating. For example, one could say, I only annotated cells that were in focus in my image. And then we have several modules that try to establish a clear relationship between an annotation and the image from which it was derived. So we have implemented both REMBI and MIFA in the bioimage archive. And users can use our user-friendly web form in our submission tool to input all of the necessary metadata for both images and annotations. And in fact, our users now have submitted several data sets with high quality annotations and with all of the information that we need to be able to reuse them. So now if you submit to the bioimage archive, the data is going to be findable. As I mentioned before, we give unique identifiers to each one of the data sets. We also provide DOIs. So you could always add the accession number or the DOI to your paper, so other people will be always able to find that specific data set. We also allow search in our website, but also within our datasets. And importantly, the data is also accessible because now you will be able to download it using different ways. So you can use direct download for the whole dataset or specific images. And for large datasets, you can use FTP or GloBIAS. And importantly, all of our datasets are under open licenses. So you can always actually access this data. And that also enables reuse because again, you can actually use the data, but also you will have all of the metadata that is needed to be able to understand the data set. But we still haven't talked about the I in FAIR, which is interoperability. And this part was trickier for us because, as I mentioned before, biological images and annotations come in lots of different formats. This is just our browsing webpage. And what you can see here is the top 10 file formats that we have in the archive, or we had at that time. And you can see that it's very varied. And some of our formats are actually proprietary. So it makes quite hard for our users to be able to work with these images, because in many cases, they even have to convert the images to an open format to be able to use them. And it made it difficult for us to make these images visualizable in all of these different formats. And at the same time, we acknowledge that downloading a dataset just to see if it was useful for you to use is not the best way to work. Importantly, because these datasets can be very large, so a lot of people cannot download a huge dataset and start working with the dataset locally. So an answer to a lot of these problems came in the form of a next generation file format called OMSR, which has been developed by the Open Microscopy Environment community. And this format relies on chunking and pyramids. So the idea here is that when you save an image in OMSR, you are going to have your original image with the original resolution, but then several subsample images are going to be saved at a lower resolution. So when you first open the image, you're going to see the lower resolution image. And then as you zoom in, you will have access to the higher resolution version of the images. And you can do that really fast because you can access the specific chunks of the image that you need to see rather than having to access all the whole image all the time. So this is a format that is cloud ready, and we decided to start using it in the bioimage archive. For that, what we did was to allow our users to submit in any format that they have, and that's something we will always do to try to facilitate as much as possible submission to the archive. And then what we are doing is to convert a subset of the images of all datasets to OMSR, and then we stage them into fast S3 storage. So with this, we are allowing streaming access to our images in a single format. And this is what we have been using to create our new website, which you can access here. We have this website running in parallel to our older or current website. This is just an alpha version. But what you will be able to see here is for our data sets, you will be able to actually explore the images in browser. And I will show you some examples now. But this idea of making the images explorable also was the stem or the idea behind our AI gallery. So our AI gallery is a part of our website that allows you to either explore AI-ready data or kind of like look at AI-ready AI data together with models. So I'm going to start with the first menu that we have here, which are our AI-ready data sets. Here, what you're going to be able to see is a collection of curated data sets that we have prepared in a way that is particularly useful for AI scientists. So, for example, if we look at this data set here, the first thing you see is an image and an annotation for this data set. And then instead of having all of the metadata that you have in our normal study pages, you will be able to see only metadata that is useful for AI scientists. So when we talk with the community, they told us, I don't need to know about how a sample was prepared for imaging, but I want to know about the license or the annotations that are available to me. And importantly, you will be able to look at the images in browser, as we can see here. This was one of the images that we converted to OMSR, and it's a time series. So now I'm moving through the different time points. And we can also adjust the contrast, as I was saying before, and zoom in and out really quickly. You can also look at this image using different community developed tools. And also you can copy the S3 link to this specific image and use it in your cloud-based analysis. So, so far I have shown you that if somebody submits images and annotations to the bioimage archive, they are going to be findable, they are going to be accessible, they will be reusable because we have the correct metadata for both images and annotations. And importantly, because we convert the images to OMSR, now they are also interoperable. And we are also curating AI datasets to be especially useful for AI scientists, and we made them AI ready. So the next step for us was thinking, okay, so now we have interoperable data. Could we start using it together with models that are also interoperable? And these models are, for example, the models that we have at the Bioimage Model Zoo. So we had a talk about that in the previous seminar in this series, but the Bioimage Model Zoo, if you are not aware of it, is our repository for AI models that is also open. So we can use those models with our data, and we start doing that in a model comparison platform. So those are the two other options that you can see in our AI gallery. So the idea behind these two options is the same. So we have a bunch of models from the Bioimage Model Zoo and a bunch of datasets from the Bioimage Archive, and we deploy the models on our data. But the two different options are catered towards different public slightly. The first option is data-centric. So what you will see here is for a given dataset, how different models have performed. And the idea here was to help people or scientists that want to start using AI models, but maybe they don't know where to start. So they can go to the gallery and look for datasets that look like their own datasets and see what models may be more suitable for their purposes. And the other option here is model-centric. So here what you're going to see is different models from the bioimage model zoo, and you will see how they perform for different data sets. And what we were hoping to achieve here is that for model developers that submit to the bioimage model zoo, they will have a chance to look at how their models perform in lots of different data sets. So it will help them to see if their models may be suitable, for example, for transfer learning. So let's look at it a little bit. This is the model-centric view. You can see when we open it, different models from the bioimage model zoo with a little bit of metadata in each one of the cards. And if we look at a specific model, you will see an image that will tell us a clue about what the model is doing, in this case, nuclear segmentation. But of course, you have metadata there to explain a little bit what the model does. And because this model was used in lots of different data sets that actually have ground truth annotations, we could compute different benchmarking metrics, as you can see in this table. So these are the benchmarking metrics for different data sets. And we can actually look at what data sets this model was better at segmenting. Apart from the benchmarking metrics, you can actually look at the outputs from the model. So if we go down, you can actually see in this column here, the original images. These are the reference annotations or ground truth. And here in the right, you will see the outputs from the model for these different data sets. So we can also visually see how the model did. And now this is a data-centric view. So this is a specific data set. We have the image and the annotation here. And again, we have a little bit of metadata, including how the annotations were created. Because this model, this data set, in particular, had ground truth annotations, we can look again at benchmarking metrics, in this case for two different models that perform the same task. And we can see which one of the models was better at segmenting this specific data set. And again, we can look at the outputs from the models for this data set in particular. But we wanted to have a very kind of broad or lots of different types of data sets to give our users a better chance of finding images that were similar to their own data. So we also deploy the models in data sets that didn't have ground truth annotations. For example, this one. So in this case, we don't have benchmarking information because there are no ground truth annotations, but you can look for this specific image, how different models perform. And if you have an image similar to this one, you will be able to choose which model was better, at least to your eyes. And we can do the same thing for models. So this is a model that was developed to segment cells of Arabidopsis thaliana ovules. But we deploy it to be used in images that have lots of different cells from different tissues, different morphologies, and different organs. And you can see how, for example, this model performed relatively well for all these different cells. So whoever developed this model could say, okay, this model in particular could be used maybe to do different things other than segmenting of Arabidopsis ovules. So hopefully, we are hoping that this will be a helpful platform for both model consumers and model producers alike. So just to summarize, I discussed with you what we have been doing a little bit regarding data for AI4Life. First of all, I talk about the MIFA guidelines. And I didn't have time to talk about that today, but the MIFA guidelines go beyond metadata. So actually, MIFA stands for metadata, metadata incentives, formats, and accessibility. And in the manuscript that we wrote for this, we give guidelines for all of these different aspects. And this is going to be published hopefully soon in Nature Methods, so you will be able to read more about this if you're interested. We also look at our AI-ready datasets, which are catered towards AI scientists to make them easier to work with this type of datasets. And finally, the benchmarking platform that, as I just mentioned, is catered to both model consumers and model producers. And with that, before I finish, I just wanted to mention that if you want to learn more about AI4Life and if you want to participate a little bit in our community and be part of it, you can join us in Helsinki for our community event, which is going to happen in May 27th and 28th this year. You can register here. The registration is open. And you can participate on the event both in person and you can also do it online. So if you are interested, please register. And with that, I want to thank the AI4Life consortium. Everything we did was really in close partnership with all the other institutions. Of course, I want to thank the Bioimage Archive team, which is led by Matthew Harley. And pretty much everybody in the team has worked in the things that I showed you today. And with that, I just want to thank you for your attention. And now I will take any questions if you have any. Thank you very much.
Thank you, Teresa. So we have time for a few quick questions. And if you don't want to speak online, please write them in the chat and I can read them at the end. So is there any question for Teresa? Okay, if not, maybe I have a quick one. So if I have a model and I would like to publish it, would you first suggest me to make it ready for the zoo, bioimage zoo, or can I also somehow connect the results of my model to the images without having to go through the process of...
You mean like for the platform that I was showing?
Yes, yes.
Yeah, so for the moment, we are only connecting with or integrating models from the Bioimage Model Zoo, because the advantage that we have with that is that they have been standardized in a way that it makes it easy for us to work with them. So I don't know if that will change in the future, but for the moment, it's only models from the Bioimage Model Zoo. So if you want to have that, you will have to first submit there.
Yeah, and I think there are a lot of advantages we have seen in the previous presentation, previous seminar. So if you want, there will be recordings of the previous seminar, how you can standardize your model for the bioimage zoo. And so if there are no more questions, we can move to the second presentation by Estibaliz.
Yes. Okay, so I'll share my screen. Can you see it?
Yes.
Yes, okay. So let me see if I can do the pointer thingy. Yes, okay, awesome. So thank you very much for letting us, the AI4Life team, be presenting in GloBIAS. It's actually very cool to show all the things that are happening behind. And thank you all the people connected there to listen to us. And of course, I think we don't need to say it, but if there are questions that arise after our talks, you can always reach us in the AI4Life email account or personally. We are very active in social media, in GitHub issues, in the ImageSC forum, and so on. So today, before starting, I would like to mention that this topic, pitfalls and perspectives about data and results quality in the deep learning era, is something that I try to build a more general presentation. So many of the works that I will present, I was not personally involved in, so I was not a direct contributor, but I think it's nice to take these considerations when you work with any sort of deep learning model or method. And also, I try to put it in a way that we can show what we've learned during these years of sharing models from the zoo and from the questions that we get in all the talks that we give. So if there's anything that is not clear, please don't hesitate to ask. Okay, so the one of the challenges, I would say, when we try to provide these models to the community, right, through the bioimage models too, is, well, researchers, they need to choose a model from our website. I will assume that you kind of know what is the zoo. If you don't know, Anna Kreshuk-Kanway gave a talk in the previous series, and I think it is in YouTube available, so you can always go listen. But So let's say that you go to our website. This is how the new website will look like. You need to choose a model, and once you choose, there's the question of how do I know if it works, right? Because this model was pre-trained on some data. And there are cases in which it is obvious, like here, right? This model is not working, and this one seems to be working well. So this kind of question, it's a very simple question and very difficult to answer. And of course, we are not the first ones that had to face this kind of challenge. And that's why I would really like to bring on the first hand this very impressive contribution from I think I counted today is around 70 authors. Now it is in Nature Methods, and I think it's the kind of manuscript that everyone should always have in the desk when doing bioimage analysis. It's called Metrics Below Their Recommendations from Image Analysis Validation. So basically, it explains very, very, very well what are the type of metrics that you should use depending on the task that you are performing. So this is a very, very long contribution. I won't go through it, but I would like to bring the attention on the very, very simple case of segmentation. So the thing is, when we try to segment objects, we usually rely on accuracy metrics. And one of the most common ones is the dissimilarity coefficient. And as you can see in this very, very nice video from Mara, she did a plugin in Apari to very quickly test how we can kind of hijack the dice coefficient, or how it changed depending on what is the result. We can see that we can get very different values from over segmenting or under segmenting, or changing the shape of the object we want to segment. So with this idea in mind, the question of how do we know that the system is valid? Of course, we have all the accuracy metrics, right? So let's say I want to segment this longitudinal kind of, let's assume that is some sort of weird mitochondria, and I just draw a blob on top of it. If I perform many of the common accuracy metrics, like the precision, dice coefficient, index over union, and so on, it's going to be higher than 50%. So let's say that it passes the threshold of being kind of accurate. Maybe not the most accurate one, but it's kind of accurate. So then the question is, are the common accuracy metrics enough, or are they opaque to the biological kind of information that we need to recover from this segmentation? There's also a second issue that is, well, often these metrics, they require some sort of ground truth, a reference image to be computed. And this reference image is not always possible to get experimentally. So here I'm showing some images from Shaling Mehta from the CCI Biohub, where they work a lot with virtual staining. So here we have the phase contrast image from which we would like to infer all the fluorescent nuclei. And experimentally, you probably know better than myself if you do experiments, it's not always possible to get 100% of labeling, especially if we use dyes. So then our reference image is probably not enough to test whether these results are accurate or not, regardless of the method that we are using. And if we see the output, well, it looks quite accurate. This is the overlap. So then what it's extremely important, and I think we should always consider is, well, these accuracy metrics are good, but we should always take biological measures into account. And the reason is because what we want to recover from any of our pipelines is biologically meaningful data and not computationally accurate one. And there is the first bridge that we should try to build in this community. So here, getting back to the very, very toy example, yes, the accuracy metric was good, but if we start computing morphological metrics like length or diameter, the blob has nothing to do with the yellow area. So in this regards, one of the very, I think, big contributions has been the cell tracking challenge, where I've been involved. And last year we got 2 years ago, we got a publication that recapitulates what has been happening during 10 years of the cell tracking challenge. And basically, one of the, I would say, the very nice perspectives that we brought is that it's not enough, as I was saying, to provide the computational metrics, but also the biologically relevant ones and the practical ones. So what are the number of parameters that we need to tune, memory consumption, generalizability capability of these methods. And here in this challenge, and now in some others, this is kind of a tendency, we are not benchmarking the deep learning methods depending on the accuracy they get computationally, but also on how relevant or useful in terms of research or in terms of biology is the output that we get from these methods. Still, I would like to highlight, and this might be covered in the next session of GloBIAS, that we, besides having this benchmark for segmentation, we really need more standardized benchmarks that consider the biologically relevant information and the robustness for all sorts of tasks. And this can be denoising, as it's been done in AI4Life, but it can also be for image restoration, virtual staining, and what's not. So this is, we ask whether the system works, whether the system is valid, and I think the very important question that we need to ask next is whether the system will always work well. And this, I'm taking a slide from Florenjuk. Here is an example of how we can actually manipulate the output of our deep learning models by using what they call adversarial attacks. So we basically kind of tune slightly the input image to get in here, in this case, they are showing what happens with diagnosis, to get the contrary kind of response from these automatic methods for diagnosis. So although this can be like a bad or let's say a negative feature of our models, we can also reconsider this fact to build more robust evaluation metrics and use it to know whether our system works well always or not. So here they are showing the authors how you can add some sort of noise and get a completely different diagnosis from this skin lesion, right? So from being benign, it can be diagnosed as being malignant. This can also happen just by rotating our images. In the case of doing the diagnosis directly from written kind of information, the vocabulary or the semantics that we use to describe the conditions or the symptoms of a patient can also influence the output of our models. And this can also happen when we do image analysis, right? So the noise that is embedded in the systems, the variability, the experimental variability that we have, we should always try to get those into consideration to test that the systems that we are using are actually working always well. So this was for evaluation, and I'm very happy to discuss more or share some sort of nice references if you are curious about it. But then it might happen that we don't even have a model to train, right? And then we need to use. So then we need to retrain our model. We need to fine tune it. And again, we go to the bioimage model zoo or any other sort of source for models. And then the data itself becomes the challenge again. And I think from the previous talk of Teresa, you already got an idea of why this is getting so complicated. Now I will go a bit more technically into the data properties. So the question is, what is actually the network seeing? And while we know that we are providing information in this case about an eye, what the network sees is just pixels. And I think this is a very basic concept, but this is a very important concept to take into consideration when we face any of these kind of exercises of building our own deep learning based pipeline. So I'm going to show you something that happened to us in the lab. We were trying to segment this mitosis in our images, and basically we took phase contrast images of CHOs that were synchronized using a 63x objective, and we did tiling here is a 16 tiles kind of reconstruction. So if you look at these images, they are pretty. They have very good quality. All the cells going into mitosis, they are extremely well defined. You can see all the details. So for us, it was like, well, StarDist will work perfectly well here. So we annotated the data. We went through the training. I tried many, many different parameters, and I was not able to get StarDist working at all. And I was getting really crazy about it because I thought, well, my data looks very good. So why is this happening? And in reality, we were asking to the network to classify each of the pixels. So that's what we do when we do segmentation, right? Each of the pixels gets a label. And the network was only seeing this very small square. This is called the receptive field, is the area that the network has access to determine the value of a pixel. And if you look at this square, well, there are no edges. There's no way to know if this is foreground or background. So, of course, there was an issue in the balance between the resolution of my images and what I was asking the network to do. So a very easy way to understand this is with art. And this is actually a very old concept. So when we want to know what should be the perfect resolution or what should be the good resolution for our network to segment accurately the images, it would be equivalent to ask what is the perfect distance I should place myself on to really decipher the scenes of Claude Monet's painting. So these type of paintings, they are very characteristics of being like a pointing, is called impressionism. And if you are very close to the drawings, you are not able to distinguish what you are looking at. And you need to place really a bit, like a couple of meters away, to see that here you have a garden, and here you have trees, and here you have flowers, and here you have people, and so on and so on. So this happened with our images. We really need to get context with still meaningful details. So basically, we need to take distance and perspective, which is the same as saying we need to downsample our data or use lower magnification objectives. That's what we did, and then the segmentation was working perfectly fine. So we tried to extend this concept in the team, and that's how we came up with our very last work that is going to be out today in bioRxiv. I just received the confirmation email that is going to be, it's been accepted in the screening. So basically, Mariana Ferreira, who is the first author of this paper, together with Ricardo and myself, we went through the understanding of how we relate the resolution of our images with the receptive field of the network. So basically, when we have a unit, for example, we know that the amount of pixels that we'll see around 1 pixel is fixed. And then depending on whether we have a very high resolution and therefore a very low throughput, the accuracy is going to be very low, then it's going to improve as a Gaussian, and then it's going to decrease again. And this can be relatable directly not only to the throughput, but of course to the resolution and the pixel size of our images. So in Rescale for deep learning, what we propose is the following pipeline. You can always get example images from the bioRxiv, as you saw now, or just run very, very preliminary and few experimental acquisition, go through the grid of downsampling and upsampling, basically rescaling your images, train for few epochs some of these networks, and compute the accuracy, the morphological metrics, or any metric that is important for your research, and the throughput, and then choose what is the optimal for you. So then what we hypothesize, or we kind of have the intuition, is that there's always an optimal, and then you can optimize your imaging parameters. So we run the experiments with a few data sets, and we saw actually that we can recover this Gaussian kind of distribution from the accuracy metrics, and there's always an optimal. And actually, although there's an optimal, you can play around with different resolutions and recover, in this case, the diameter of this E. coli bacteria. So this becomes interesting because then it means that for every single deep learning method, all the ones that we have in the zoo and all the ones that you will train, and including foundation models, if you're interested, go to the paper, you can always find an optimal image resolution, and usually is not the one that is the best one in your microscope. So this is very important because it means that we can get cheaper data and faster data. So we went through this concept with the staph, and we did a very simple experiment of having staph bacteria treated with an antibiotic that doesn't allow the bacteria to divide. So then we have larger bacteria, actually double the size. And we trained different methods at different resolutions. Many of them were accurate. And actually, with all of them, we were able to recover the phenotype. So this means that we can really push the limits of the resolution to exploit these deep learning methods. And also, it means that sometimes when our methods don't work, it's probably because the data is not adequately given to the method. Of course, there are more works related to this. We have this very nice perspective from Stojiljkovic that they are showing how we can actually configure our microscopy acquisition depending on what is the information we want to recover, whether it's the mitochondria or whether it's the nuclei, and how some of these features are preserved along the different objectives. So this is cool, okay? We know that we need to be aware about what the network is seeing, but not only what is seen for each image, but what is seen at each time. And here we are speaking again about variability, which is one of the very, very, very intrinsic and natural features of microscopy imaging and bioimages. So the challenge is that this variability is huge in microscopy. And one of the issues that we have is that experimentally it varies. So here I'm showing you two CHO images taken with transmitter light microscopy, same microscope, same experimental conditions. We are just changing the person who is taking the image. And you can see that they are very different. Actually, they even look like different phenotypes. Here we have elongated cells. Here they are like a smash egg, right? If we look at how they look in different modalities, where here I'm showing you mitochondria with different microscopy modalities, they are all different. So how do we deal with this? Well, of course, there are many complex works on domain adaptation, but one of the key components that we should always take into consideration is normalization. So while in computer vision and in general, people go through the standard and mean normalization, this doesn't always work well in microscopy. And first of all, if we look at these two images, well, they are very different. Here we have many cells. Here we have a few. Here there's a lot of background. Here there's no background. Here there is illumination artifact. There is uneven illumination, very common from bright field microscopy. Here there's almost no illumination artifact. And if we look at the histograms, they look the same. So the common approach of looking at the histogram and normalizing the data here won't work well and will cause you many troubles if you normalize the data like that and then you try to train your method. Then we have the commonest scenario of having uneven fluorescence intensities due to photobleaching, but also due to the natural standard deviation when you dye your samples. Like you can have some cells that express a lot of some sort of fluorophore and others that express less. So of course we can go with the percentile normalization. This is great, and actually this may be good, but then the question is, well, you are removing some biological condition of these cells. So what about if I want to recover the molecular information? We can even go one step further, and this was shown in Martin Weiger's work in CARE. If you don't take care of the normalization in the input and the output, and you don't account for having this background, well, your results might be badly expressing what you have in your sample and may, for example, enhance the signal in the background and so on. So these kind of normalizations is not as straightforward as given by default by just normalizing each of the patch in the networks. And some strategies that you can have, well, there are the common ones here with the 0-1 projection, mean standard deviation, percentile projection, but these should not be done patch-wise, but you should always consider your entire population. So maybe you need to normalize against the entire tissue, against the entire experimental replica, against the entire acquisition experiments. So you should always consider how is your data, what is the source, what is going to be analyzed in the future, and what is the variability that your network will see. Because although you have all these priors, the networks, they don't have. And here I want to highlight 3 works that I really think they do a very nice work in terms of normalization. It's usually in the supplementary material. So this is my favorite one, where they describe a pipeline to normalize against replica, acquisition experiments, and whatnot, because actually they were trying to recover the true intensity value and not just to segment the images. Of course, the work from Martin Weiger, that is from where we took this one, and a recent work from Anna Kreshu, where they are really showing how when you work with very, very large tissues, you should consider all these aspects. So far, I spoke about technical details and how does this relate to the work we do in AI4Life? And what we learned, I think it's obvious. This kind of talk, I think years ago, was not the kind of talk that we were able to give because many of these aspects were kind of new. And where we are now. So far, like we mentioned, that we need fair data and AI-ready microscopy data. We need guidelines to adequately validate our results and report these results. We did better data curation practices. So when we normalize the data, it shouldn't be just a simple 0-1 normalization. And we need to really better understand the data model research axis that I think we are here at the moment in this part of the bridge that we are trying to build. And all this relies in something that is extremely important, and this is where we are doing the job, and is on developing standards for all this to be, for all of us to be in the same page. So very briefly, I think Anna and Wei presented the bioimage model too, and how we are building these standards in the previous talk. So one of the very key components of the work we are doing is to define a resource description file. So basically the model metadata and the image metadata. So in the model metadata, yes, we need to know how the input and the output shapes there are, but also what is the receptive field. So here we are starting seeing the words that I was mentioning before. What is the normalization? So it is true that we are not as advanced as we should regarding tissues and whatnot, but we have some kind of pipelines defined for this normalization, and this is always an open discussion. Post-processing, how the tiling should be done, and there are all the works that are coming out now because all these questions appear when we are working with all of you trying to provide these models for the community. And also we have some information about the biological data being analyzed. And this is again important, I think, as I already tried to convince you so far, that is not only about pixels, but it's also about the physical information that is embedded in these pixels and how we provide this information. So all this is in the specs or the resource description file. It's a text file that we have programmatic tools to access to, so we can build these pipelines by accessing something else besides the input shape. And this is where we are building the connection, of course, with the bioimage analysis open source tools and the community partners. So because I don't have more time, and of course I don't want to get you bored, if you are really interested on how we are building these standards and what are these standards about, I would like to re-highlight, as Teresa did, the community event that is happening in Helsinki in May. But also, if you are really, you cannot wait to know about more, to know more about this. Tomorrow, we are again giving a series of talks on AI4Life in the Image Data Community Day. So registration is still open, and you can listen more about this. So the take-home message from this is that yes, we need data standards, but we need improved ones for AI, and we are building them, but more work should be done here, and any contribution is more than welcome. We need robust and transparent validation that takes into account reproducibility and also the trustworthiness in terms of biology and in terms of the knowledge that we are extracting from these models. So for this, yes, we need the validation guidelines, but I think the community will really benefit from further standardized benchmarks, and not only for segmentation. And we need to start thinking that the optimization from all these pipelines, it's not coming from the end solution in the model, it's really coming from the data acquisition. So here, I think we really need to start switching our minds also in the computer sciences field, and we need to start thinking about hardware-aware or physics-informed AI models, or at least how to properly treat this data, or know more about the data before analyzing this. So with this, I would really like to thank Enrique's lab, where I'm doing my postdoc at the moment, and Mariana also for the work I presented today, and Ricardo. We are also community partners in the AI4Life, and of course the entire AI4Life team and the people behind that. Maybe they are not in this picture, but they are always contributing to the work that I show. So with this, I'm very happy to answer any question.
Thank you, Estibaliz, for highlighting so nicely the many challenges that we have, not only when we build, but also when we want to use models. So is there any question from the public before we close the seminar? Kesavan, please.
Hi.
Yeah.
I had a question regarding context. So, I mean, when a microscopist designs an experiment or a biologist designs an experiment, there is a hierarchy of context, right? Like, for instance, the domain knowledge and the specific project, and then an experiment itself. And on top of that experiment, maybe even what fluorophore you're using, and whether it's a live cell or a fixed cell, all these kinds of like context that adds towards the final image, the decisions that kind of lead up to the final image. So how does one kind of bring that into the image analysis domain? Because when we think about bridging biologists to the computer scientists, and in the computing, there is this cognitive computing as well, which is where all these AI kind of things comes in. And but the cognitive aspects of biologists is not entirely carried forward with the images. So what are your thoughts about that?
Well, I think that's what we call metadata, and is what Teresa was describing also when she was explaining how you should document your images. So I think there's a very big effort on these bioarchive data repositories to actually gather all that information. So I mean, for example, usually microscopy images, you have the image, the raw data, right? And you have the metadata, and you have all the setups from the microscopes and so on. What I think we are missing is some sort of format, and I think Teresa can give more input than myself there, where we have that information standardized in a way that we automatically read from the model. Also, so my experience is that the people the people in the zoo and myself, we are very aware of how all those parameters affect the models. But usually computer scientists, they want to approach the highest benchmark with the models and nothing else, because what they do is developing models. So maybe when we do the benchmarks, that's also the type of information that we should evaluate and that we should provide, and we should highlight that it's very much needed. I'm not sure if I am answering your question, or Teresa, you want to add something there?
No, I think what you just say, it's I think like those type of metadata can be definitely, like if they are properly input in a repository, then it will be much easier to work with them in the future, and then obviously keeping track of them.
And I also had a short question. Like ultimately, the goal is biological insight. If you can hear me well. So, but I do not see the insight generation process being incorporated into all these pipelines that we discuss.
But okay, so maybe because we didn't explain very well the zoo, so one of the key aspects of the bioimage model zoo, and what makes it really different from other like Hugging Face and what's not, is that the model format is not only about the specs in the model, but you should always provide some minimal information about the images. So besides providing like the cover, where you very clearly show the type of images you're analyzing, you should describe what was the data used to train the model, for example. So there, the ideal kind of a scenario for us is where when you upload a model that was trained with some data, and you make this data publicly available. And this data ideally should be in a public repo like the bioimage archive, where all these details that you mentioned, like the type of sample, the experimental protocol to obtain the data, the microscopic parameters and so on, are very well detailed. So there is where you have the connection. Like this model was trained with this data that has these very like experimentally related information. And that's what we are trying to build now. So of course, many models that we have in the Zoom, maybe they are not perfectly documented, but during the reviewing process also, when researchers upload their models, we test that the format is correct, and inside the format, all the mandatory fields related to the data should also be correct. But we also kind of try to push a bit researchers on doing this kind of linkage. And of course, things need to be open. So yes, that's part of the effort. Let's see if we manage to make it to succeed. Okay.
Sorry, I just wanted to clarify, because the question is what next? Because once you have all that information, that's when the insight generation process starts, right? So what are the algorithms for insight generation? That's the question. But thank you so much.
Thank you. Even if you have only a short question, because
yeah, I'll try to be short. Do you hear me?
Yeah.
Hello. Nice. Okay. Thank you very much for nice presentations. It will be very short questions, but two, if you no, please. So first one, concerning the biomodel zoo. Sorry, it's just a little bit long, the name. I was just wondering, I promise I will see the video, the previous webinar. I didn't have a chance to check it out. The only thing which I wanted to ask, do you have any framework which is in the build for the fine-tuning models? Because you have a lot of models. I see most of those are UNets in different tastes. And still people can have some, desire to do some fine-tuning just in the platform. So is it possible to do it right inside the platform, or everybody should do it himself?
Yeah, that's a very good question. So at the moment, inside the zoo, it's not possible. That could be ideal, and it's one of the ways to go. So one way to do it is by using like zero-cost deep learning for microscopy, BioPy, Ceramics, so some of the software, some of the community partners that they provide user-friendly notebooks to fine-tune these models, so they can load the model and then you can run the fine-tuning for some of them in Google Colab or in your local machine. And they provide like user tools to do so in a user-friendly manner. Another way, if you want to go more programmatically with more complexity, you can always use our example notebooks, both in the bioimage.io GitHub account or in the deep learning for MIC everywhere software. We provide some notebooks where we show how to go with the code, load the model, and fine-tune some of them. Still, if none of these approaches work, in the zoo, when you go into the model, you will see that there's a chance for you to open an issue and to make questions. So you can always use that to say, yeah, I'm really, really interested in fine-tuning this specific model. Can you give me some hint, or is there any code that I can reuse? Usually the models, they are linked also to the original publication, where usually they provide the code, or they are linked to some GitHub repo that you can follow. So we try to make all this linkage to guide you through the process.
Just maybe small question. You mentioned that I can find this description of the notebooks, and those notebooks are all there. So all these manuals for the fine-tuning and stuff, I can find it there.
I think I didn't get the question.
You mentioned just several platforms which were in such speed that I didn't manage to conspect.
Yes. So if you go to the zoo and you type PyTorch notebook fine-tuning, it will pop up a notebook.
Okay, beautiful. Yeah.
For fine-tuning. But otherwise, if you follow the examples from our coding packages, you can also find it there. And if you don't find anything, just drop a question in the forum or in GitHub, and we will answer you if you still.
Okay, thank you.
Struggle with
Yeah, thank you.
Sorry.
Maybe. Yeah. Okay.
No, sorry,
okay.
we don't have any time left for the discussion, and I would like to thank again Estibaliz and Teresa for this nice presentation, and see you at the next seminar. Thank you.
Thank you.
Thank you. Bye.
Bye, everyone.
Bye.